Morning, everyone. It's a great pleasure to be able to introduce our next speaker, Andrew Leduc. Andrew comes to us from Northeastern, and he's made important contributions to moving single-cell proteomics forward as a field. Andrew developed an approach that massively scales up our ability to prep samples for single-cell analysis, an approach called NPOP, that allows us to collect and prep thousands of cells in a single day. Really exciting development. And Andrew's used that approach and other approaches to make important discoveries in the field of single cell biology, one of which he'll talk about today. But with the time that I have to introduce Andrew, I also want to highlight another thing that I think is an important aspect of Andrew's character, which is his willingness to help others get into the field of single cell proteomics. So Andrew has traveled around the country, around the world, training others in how to use NPOP. He's readily shares data. He's actually come on site to our lab and helped us by sharing data, by helping us learn the protocols that he's developed. And I think that's really important because single-cell proteomics moving forward as a field depends not just on individuals making great discoveries like Andrew's done, it also depends on empowering others to innovate and to make their own discoveries. And Andrew's done a great job with that. And in doing so, he's really established himself as a great leader and a colleague in the field of single-cell proteomics. So please welcome Andrew.
Thank you. Thanks so much, Malin, for that really nice introduction. I think this is my 7th single-cell proteomics conference. The first one I was an undergrad coming just out of undergrad in chemical engineering, and I think I thought proteins were mostly a dietary supplement. So hopefully I seem slightly more knowledgeable in my talk today.
Go.
So today I'm going to be telling you a little bit about our recent work quantifying modes of regulation for protein abundance in single cells from primary tissues. Get this guy off the screen. You move that box. Maybe it won't get in the way too much. All right, so multicellular life, as you know, has this amazing ability to create a diversity of cell types from a common set of genetic information. And of course, these diversity of cell types arise because these cells regulate their genomes to express different subsets of proteins and express these proteins at different levels that give rise to these diversity of different functions that these cell types have. So it goes without being said, if we want to better understand the diversity of cellular phenotypes that arise in multicellular organisms, we'd like to be able to measure the proteins that these different cell types express so we can learn a bit more about their function. And one of the early technologies that allowed us to get an idea of what proteins are expressed in single cells for thousands of genes across thousands of single cells was, of course, single-cell RNA sequencing, which lent us insight into the transcriptional processes that lead to the production of different proteins that allow cells to carry out different phenotypes. Yeah. Sorry.
Hold on one second.
And more recently, of course, single-cell proteomics has emerged, and I'm sure you'll hear a bit more about the development in of these technologies from others in the talk, and they're technologies that I've been lucky enough to have a chance to work on in my time in Nikolai's lab over the past 7 years. And of course, while it's great to measure protein abundances to get a better idea of how these cells work, we also want to know the methods these cells use, the ways in which these cells are able to regulate the protein abundances. And while this is interesting, not just because it helps us learn about the biological circuits that control cells, of course, this information can also be practical if we want to manipulate cell states and devise treatments. As we just heard in the last talk from Galit, that if you wanted to modulate p53 levels, you might be interested in modulating them at the level of protein degradation, where the pathways are really controlling the abundance of this p53 protein. And if we think about methods that we are really commonly used to study the ways in which protein abundance is regulated, a lot of these work at the level of methods that kind of control transcriptional processes. So we have methods to explore kind of transcription factor binding and DNA interactions and methylation and histone modifications. And ultimately, these are a really important set of regulatory processes that give rise to differential transcript abundances across single cells, which ultimately influence the proteins they produce. But as we know, the regulation of protein abundance doesn't end at transcription. There's many translational processes and regulation of protein clearance that play a role in kind of developing and contributing to the ways in which the proteome is shaped across different single cells. So if we want to get a better idea of what the contribution of these kind of post-transcriptional processes are, translation and protein clearance, to determining protein levels, one approach that we could take would be to sequence single cells at both the level of mRNA and at the level of proteins. And recently, this work was done in our lab led by Sa Khan, where he took cells from the human testes and measured single cells of the protein and at the mRNA level and integrated this data. And he could use this data to assess the extent of discordance between protein abundances and mRNA abundances in single cells from a given cell type. And from these measurements, you can kind of infer a difference between the protein and mRNA levels and determine the amount of post-transcriptional regulation that's kind of going on past just the level of mRNA abundance. But you might be interested in taking it a step further and narrowing this down and learning whether it's really translational processes or processes in regulating protein degradation and protein clearance that contribute exactly to the discordance that you're seeing between these mRNA and protein abundances. And a really useful technology for being able to do this is the metabolic labeling of proteins with heavy amino acids. And this technology works by essentially at time point 0, you spike in these heavy amino acids and proteins are slowly synthesized. And you can measure these on the mass spectrometer with these kind of peaks that pop up, the number of Daltons heavier for They're being cleared as the cell kind of divides more rapidly than the protein half-lives allow. So to try and answer this question for tissues in the kind of a wide array of tissues in the human body, we applied this metabolic pulse labeling strategy to in vivo in mice, which you can do simply by, instead of kind of changing the cell culture media to have heavy amino acids, you can just feed the mice this different food that has these heavy amino acids, and this allows you to measure these protein synthesis and degradation rates in this kind of wide array of tissues. And when we measured synthesis and degradation rates in the brain, one of course, a tissue where there's really slow cellular turnover, of course, neurons mostly kind of never dividing in the life of an organism. What we saw was that there was actually a really strong relationship between the rate at which proteins cleared and the kind of overall abundance of these proteins in the cell. And thankfully, since we measured other tissues that had a kind of variety of growth rates, when we looked at these trends as a function of the average cellular division time of the tissue, we found this strong relationship where the faster cells divided in tissues like kind of the bone marrow, we found very low control by protein degradation, and that kind of gradually increased as the doubling time of the cell got longer. And here I just want to point out kind of the type of regulation we're talking about is within these samples, the ways in which the abundance of kind of different proteins are controlled within a cell. But of course, it's also important to think of the kind of converse, which is for a given protein, how is its abundance controlled kind of across different tissues, across different cells by these kind of mechanisms of degradation. And what we saw was when we compared the rates of protein clearance for two tissues that kind of divided more slowly, the brain and the pancreas, what we found was that kind of off scatter points here are going to suggest differences in degradation rates between these two tissues. And when we compared the fold changes for clearance rates to the fold changes for differences essentially in protein abundance between these two tissues, we also found that the relative differences in protein levels for the same gene were kind of strongly controlled by differences in protein abundance. And if you want to hear more about this kind of brief story on the role of kind of dilution in determining the ability protein degradation has to determine protein levels, you can read a bit more about it in our recent paper. So back to single cells, are we also able to measure these mechanisms and also kind of transcriptional mechanisms in single cells and not just at the whole tissue level? So we recently set out to do this again, and we were able to kind of feed these mice this special diet, and we were here analyzing cells from primary kind of murine tracheal epithelial samples. And so we took certain cells for proteomics, and then from kind of the same type of dissection, we took cells to sequence the mRNA through just 10x genomics. And for the protein prep, we utilized kind of these multiplex mass spectrometry technologies where for each run on the mass spec, we were analyzing 2 single cells at a time, and we achieved this with kind of these heavy isotope tags that we are able to kind of put onto the end of the peptides we're analyzing. And when the peptides come into the mass spec, that you kind of get this trend where you see a peak corresponding to the first cell, which has its light and heavy component. And then you have a kind of this 16 Dalton separation, where now you can kind of see, again, the differences from the second cell. And these technologies allow us to kind of increase the number of cells that we analyze and infer information kind of from more cell types. And so after collecting this data, we generated a data set that had about 4,000 single cells at the protein level and about 36,000 single cells at the mRNA level, with roughly 2,000 kind of shared genes quantified between these. And when we looked to try and see the cell types that we were identifying in these two samples, what we found is that there was kind of, of course, as we expected, we found kind of similar cell types between the two samples. And we wanted to compare to make sure our assignments were kind of confident and similar between these two data sets. And so to do that, we can just compare the relative levels for kind of like cell types at the protein and mRNA level. And of course, mRNA is a really important part of the regulation of protein levels. So we expected to see some strong agreement. And here you're just looking at, I'm just visualizing two cell types, and we can see is kind of the relative average mRNA level and average protein levels across cell types kind of agree strongly for the two cell types that I'm showing. And we can extend this visualization to all the different cell types that we are able to measure. And on the diagonal there, it's kind of the agreement between the kind of the average protein levels within a cell type at the protein level and at the mRNA level. And this kind of gave us confidence that our integration was faithful and that we were kind of comparing apples to apples when we were comparing two cell types at the protein and the mRNA level. We can also look at the kind of reproducibility of the measurements we're making. So here we're just taking kind of split pools of cells from within a cell type and comparing kind of the agreement. And we see that kind of we have strong reproducibility for our mRNA and our protein measurements. But again, we wanted to be able to kind of measure these translation and clearance rates for these proteins as well. And again, so we can apply this metabolic labeling technology to also infer kind of the degradation rates. And then with this equation, with these kind of 4 parameters, we have the leftover variances going to give us the ability to understand the translation rates in this for these proteins as well. And so we can also assess the reproducibility of the degradation or clearance measurements, and as well as the translation, which is going to be defined kind of as translation number of molecules produced per copy of mRNA. And so in a given cell, we can quantify these degradation and translation measurements for a little over 500 proteins, and we can quantify abundance measurements for about a little over 800 proteins per cell, with the total set being about 1,500 for degradation and translation measurements and a little over 2,000 for abundance measurements. And you might be wondering about the variation in these distributions of box plots, why some cells might have more proteins quantified than others. And a lot of this comes down to kind of the size of the cell. For bigger cells, we're able to kind of quantify more proteins. And you can see here this really nice scatter between kind of the volume of the cell, which we can measure through our sample prep method, and the total amount of protein that we're measuring, which kind of gives us another layer of faith in the reproducibility of our sample prep for these samples. And so earlier I was talking about the ways in which kind of growth played a role in determining the influence of clearance in these cells. So ideally, we would also like to be able to measure directly the rate at which kind of cells are dividing in these samples. In addition, so we can kind of disentangle influences from protein degradation and growth kind of due to dilution. And so this growth, again, is just kind of proportional to the doubling time of the cells. So really, if we measure the cell cycle time, this gives us the ability to infer kind of this growth parameter. So how can we kind of measure the cell cycle time for these populations? And when we look at the turnover of histone proteins from these samples, what we can see is we get these actually really discrete distributions, because histones mostly are not really degrading very quickly, except for that one kind of point in cell division where they have to kind of double the amount of histones produced. So it leads to these discrete bins here, kind of reflecting cells that have divided once or twice. And we can use this information to compute the doubling time or the growth rates of the cells. And you see that there's a nice amount of kind of variation across these cells, with some cells like these basal cells, which are kind of these stem cells that replenish the epithelium and the dividing quite quickly, where other cells like these tissue resident chondrocytes dividing quite slowly. And so what effect does this have on the kind of differences in measured clearance rates? You can see that when we compare cells that divide quite quickly with cells that divide quite slowly, it kind of shallows the slope of the measurements between the clearance rates. And essentially what this means is that for cells that divide more slowly, you're going to have a lot more variance in the clearance rates compared to cells that divide kind of quickly, and that high growth term is going to kind of limit the cell protein to protein difference in clearance rates that can kind of be obtained. And you see that kind of relationship as you compare cells that kind of have a smaller growth differential, that slope kind of narrows closer to 1. And another way to kind of visualize this is to kind of just plot the variability of these clearance rate distributions as a function of the growth rate. And you can kind of see we have this nice trend. So, the question is, do we see these same trends recapitulated in the single cells that we found in the tissues, where cells that kind of grow more slowly have more control across proteins due to degradation? And what we can see in the cells that chondrocyte cells, which are some of the slowest dividing ones we measure, there's quite a strong association between clearance rates and, again, this protein abundance, much kind of more so than that 5% that was originally measured in these rapidly dividing cancer cells. And so we can see this trend again, we can see this kind of similar trend with the correlation to the growth rate of the cells. And the question you might have next is, what processes are kind of compensating for this, picking up the slack, and for cells that divide faster, that are contributing kind of to a greater extent to the amount to the regulation of proteins. And what we could see is, in particular, the kind of translational control per mRNA seems to kind of increase considerably with the rate of the cell, and potentially making up for that variance that's kind of taken away as the differences in protein degradation throughout the cells. And of course, we didn't just want to measure kind of within a cell type the protein abundance control. We also wanted to look at this across cell types. And so one way we can do that is for a single protein, we can plot the change in kind of concentration across different cell types. And so you can see the mRNA in the black line, and you can see the protein levels in the orange line. And here we have quite strong control for this protein by transcription. And so we can compute a correlation from this trend, and we can kind of visualize that distribution of correlations. And we can see that on average, this trend is quite strong. But of course, for other proteins, we see that there's kind of a less degree of discordance, and we might kind of suspect that other modes of regulation are important for this protein. We can also visualize the same trend for degradation rates. Here, I've plotted kind of the protein half-life in blue and not the degradation, because the kind of half-life is going to vary with the inverse of the protein. So, but the half-life, think protein degrades more slowly, you'll have more of it. So you again see kind of a strong correlation for this particular protein to the protein half-life. And then when we look at the distribution, again, of correlations between protein abundance and this degradation rates, we see on average this negative trend, which suggests that maybe to a lesser extent than the mRNA, but there's still some kind of control going on by protein degradation. And so for this particular protein, we can, it turns out that protein that we just visualized with poor concordance with the mRNA is strongly controlled by degradation. And so you can kind of see how we're able to unpack these modes of regulation for kind of individual proteins that might be regulated similarly across single cells. And we can visualize this not just for one protein, but we can kind of see this trend generalized across the whole proteome. And what we can see essentially is for proteins that have high negative correlation with their degradation rates. So that's to say, as the protein degrades faster, there's less of it. We find that the correlation between the mRNA is also decreased. So kind of we're seeing this trend generalized past just this kind of one single example. But of course, we don't necessarily need to assume that this degradation, the modes of regulation, are going to be kind of continuous across all the cell types. We might have cases where a protein is degraded by degradation in one cell type, and then translation drives the protein abundance in another cell type, and transcription in another cell type. So we want to also look at these trends kind of on the individual cell type level. And so take these chondrocytes that these cells rely heavily on glycolysis. And you can see for this particular protein, glycogen phosphorylase, on average, it's about twofold higher in these cells. And so these cells are kind of they're tissue resident cartilage cells, and they kind of live in low oxygen environments, of course. So these cells really kind of rely heavily on glycolysis versus kind of respiration. So when we look at the control by mRNA, we see that the mRNA levels are kind of quite not very differential for this protein, and not kind of contributing strongly to the increased abundance of this protein. And from the previous work, we would maybe look at the difference between these protein levels and mRNA levels, and we would infer some difference, and we would say that kind of post-transcriptional regulation is happening kind of in the cell type on some level. But now we can take this a step further and we can look at the contribution of translation. And we see that that also is not strongly contributing to the increase of this protein. And we can remember that these chondrocytes, when we were kind of looking at the regulation across the proteome, we found that degradation was quite important for these kind of slowly dividing cells. And in this case, we can see that the slower degradation of this protein in these cell types allow them to have higher abundance of these proteins that participate in glycolysis and kind of allow the cells to kind of carry out this important metabolic function and provide energy to these cells. We also measured, again, these kind of basal epithelial cells. And one of the markers of the cell is kind of this keratin 5 protein. And we can see that this protein is kind of massively upregulated in these cells. It's about 6.6 on a log 2 scale, so almost 40 times upregulated kind of versus the other cells where keratin was measured in this population. And these keratin proteins are quite important for all these kind of different epithelial cells that are kind of constantly proliferating and kind of moving up towards the surface. They need to express different keratins to kind of hold different shapes. And so if we're interested in learning about kind of the regulation of this protein, we might suspect, of course, the mRNA abundance needs to be high in order to kind of produce a 40-fold increase in this abundance of these kind of keratin proteins. But it seems like the mRNA fold change is kind of not sufficient to kind of explain that really 40-fold kind of high abundance. So you might think that maybe the protein is degraded slower so that it kind of can accumulate in these cells, but we also find that that's not the case when we look at the contribution of degradation. And that allows us to kind of understand that not only is the increase in transcription important for these cell types, but also the translational kind of per mRNA translation of this protein is important to kind of allow these cells to have high abundance of this kind of keratin protein. And the fast turnover might be because, these cells kind of need to divide quickly and be able to kind of differentiate into cells and kind of lose that keratin protein so they can take on different shapes. Okay, and so with that, I think I'm about at time. I lost track a little bit, but yeah, okay. So I'll just say thanks so much to the organizers and to my colleagues in the Slavov lab, and also our collaborators at MGH who allowed us to kind of do all these animal experiments and helped us collect the data that we talked about here in this presentation today.
Someone asked a question on the chat. So thank you for the excellent presentation. Could you please provide an estimate of the minimum number of cells required to reliably annotate the population? And then
Yeah.
can you use cell markers derived from mRNA data or from?
Yeah, so there was a question online about kind of the number of cells needed to quantify different populations at the protein level and how we were able to kind of assign cell types, whether the markers were coming from mRNA data. In terms of having enough cells to kind of reliably assign a cell type, I think we pretty commonly find that we can identify these cell types with kind of quite a small number of cells. I mean, of course, you want to measure enough so that you can kind of compute robust statistics. So, as long as you analyze enough single cells to get, 10, 20 cells from this population, you're usually able to identify them. And these markers came from some at the mRNA level, but also some kind of that have been studied in measurements with there's been some that have kind of these markers have been validated through antibodies. Here you're seeing these kind of keratin 13 plus subset of these epithelial cells, which we were studying that kind of, you can kind of clearly see these markers with antibodies and fluorescent-based proteins. So it kind of came from an array of sources. Thanks.
So I think you may have just answered it, but these are hand-curated marker proteins for each cell type?
Yeah, so in the original assignment, when we kind of originally assigned these populations,
You map way back in the middle.
yeah, we had, yeah, so to be honest, there's like a host of different methods you can do to integrate these data. In the past, we've kind of relied on popular tools like Liger to do the integration, which kind of also facilitate them. In this particular case, I found that the kind of clusters were quite clear, and you could, from kind of just doing a quick literature search and seeing what proteins were expressed in the different clusters, it was easy to kind of differentiate what cell types were where. And then, of course, like, mRNA abundance, while maybe not the complete story, is obviously kind of like one of the really most important modes of regulation for protein abundance. So you should have a default expectation that there's kind of a really strong concordance between the two. And I think with these kind of integrated analyses where we're kind of taking, not measuring mRNA in the same cells as proteins, it's of course like really important to make sure that your integration is kind of reliable, because any discordance in the integration of cell types can kind of lead you to misinterpret variation. From it might just be a noisy integration versus.
Thank you so much, and thank you for persevering through your talk. We really appreciate that. Do this. So you mentioned that you can actually measure volume when you're in the process. So that potentially gives you the opportunity to then actually make very precise measurements about concentration as well. Is that something you guys are doing or can integrate? And can you tell us a little bit how you measure volume?
Yeah, that's a really good question. So doing the sample prep, we rely on this like cell sorter that's imaging based. And so for every single cell, we have a picture. And it kind of gives you this rough estimation of the diameter of the cell, and then we kind of just use that to compute the volume. I think like that's a really exciting analysis too. Generally, we have like the assumption that the concentration scales pretty well with size, but I think looking at the variation, like in general, is a really fruitful area of analysis. There's a slight question of whether the pictures that we get are like how quantitatively you can measure the volume, but I think like as that technology improves, it's a really exciting kind of way to analyze the data. Mostly I'm using it for like if there's a massive disagreement between the volume computed and the cell, you kind of can suggest doublets that you might have sorted and things like this. But yeah, it's that and like image analysis of the morphology of the cells correlated with protein levels. There's kind of a lot there for people who are more inclined with image analysis.
So this question may not exactly relevant this one, but I just want opinion. Degradations. Are you referring to the proteosomal degradation or something, a different way of degradation? And the second thing is like, even if it is a proteosomal degradation or different degradations, whether the degradation is giving degrading the protein into individual amino acids or the fragments of the like peptide remnants. If they're producing a peptide remnants, when you do a quantification on the mass specs, how do you differentiate that the peptides that we are measuring are coming from these remnants or the intact proteins? So, and also a question is like, because when you're referring, sorry,
I
want to complete the whole.
Mhm.
So, when you say protein degradation, I'm just also curious, did you ever try using any PROTAC to target a protein of interest to just compare this one with the real protein degradation stuff?
Yeah, that's a really great question. So, of course, this is like, you're getting slightly closer to understanding the regulation of these proteins by knowing kind of the main mode in which through they're regulated, be it degradation or translation or transcription. But of course, there's like a lot of different ways a protein could be degraded. And I think this is like a really powerful tool for kind of hypothesis generation for you understand that. So before you do any like perturbation experiments, I think there's kind of a lot of fruitful analysis to be done, just seeing like, the abundance of certain ligases increases in these tissues where you see the corresponding like relative increase in degradation. And this can suggest, whether it's kind of through an E3 ligase proteasome pathway, or maybe you can look at kind of different lysosomal proteins that are increasing on a lot in a certain cell type where you're seeing, faster degradation of other proteins. So I think kind of, just through like these associative correlative analyses, you can start to kind of develop more fine-grained hypotheses that will then allow you to kind of do better experimental tests to kind of exactly understand the precise mechanism through which these proteins are degraded. Through using kind of things like PROTAC or kind of even degrading certain E3 ligases that you found might be upregulated in a certain cell type, and seeing kind of how that influences these degradation rates and whether you can kind of pin down a more confident mechanistic relationship between a protein who's degraded faster in a cell type that happens to have more abundant a certain E3 ligase, and whether there's kind of a direct interaction there. So it's kind of closer to mechanism, but also maybe a good method for kind of generating hypotheses for more specific mechanisms.
All right, we have a couple online questions too, and I think what you just said is a good segue to the first one, which is for cell types that rely more on degradation, do you also see an increase in the abundance of the respective machinery? For example, ubiquitous system enzymes.
Yeah, that's a good question. Certainly, the abundance of the proteasome kind of varies a lot across proteasomal proteins varies a lot across these cells. I don't have any kind of data exactly to speak to this, but we see that proteins of the proteasome themselves are kind of controlled a lot through degradation. So I think their abundance is higher in these kind of chondrocyte cell types that divide more slowly. And I think that kind of reflects the fact that that kind of specificity of degradation are more important in regulating the proteome of some cells versus other cells.
And then the next online question is says, amazing work. You've measured protein data for 4,200 single cells. Two questions. How many proteins are you able to measure per cell? And how long did it take to measure 4,200 single cells? Is the throughput 2 cells per run with mass tags?
Yeah, great question. So the throughput was 2 cells per run. I think the active gradient was about 10 minutes. All in all, it took about kind of like roughly 2 months of mass spec time to acquire the data. And the reason that there's kind of difference in these two numbers, whether it's kind of proteins where we can measure synthesis and degradation or proteins where we can just measure abundance, is because we're only pulsing in like a heavy lysine, but we're using a tryptic enzyme to cleave the protein. So we also end up measuring a lot of proteins where we just have kind of arginine containing peptides where we don't get that kind of heavy light split. So yeah, I think there's like also exciting work to kind of pulse in other non-essential amino acids and increase the number of proteins, which you can also infer this for. And you can also use like other enzymes like lyse C, which don't always perform as well as trypsin, but might give you a slightly higher number of clearance and translation rates quantified.
Andrew, thanks for the great talk. I'm curious if you have considered looking at aging, as a lot of age-related diseases are degradation dependent or related.
Yeah, good question. So this research was actually funded as part of a grant to study cellular senescence. And we have here some of the mice are like really young, just a couple of months old, and some of the mice are kind of 24 months old. We were really excited to study this. These experiments kind of are a bit expensive. And sadly, we had 2 of our old mice that were like 24 months old pass away like 9 days into feeding them with this expensive food, which like unfortunately like lost 2 data points in the experiment. So we don't like have great statistics for looking at age with this particular data set, but we definitely plan to kind of look at this a good amount.
Other questions for Andrew?
Just quickly, how do you take into account the dynamics of lysine in the body? Do certain tissues absorb it differently, and metabolism happens differently, and how does that impact?
Yeah, that's a good question. So we're able to get essentially like there's a parameter in the equations, which is like the free pool of lysine that's available. And we can get all right estimates for this in kind of on an individual single cell level by looking at peptides where there was a miscleavage. And so they have 2 lysines. And essentially, with some probability, they'll be translated with kind of both the lysines heavy or both the lysines light or one light and one heavy. And through like these, through kind of leveraging this information, we are able to compute the kind of roughly what the free lysine pool is, and then adjust the kind of measurements to understand these rates. And one like kind of quick sanity check for us is that here you would expect kind of cells that have divided once to have like roughly 50-50, and it looks like, maybe slightly more heavy than light histones because there's probably also kind of some turnover of the histones themselves in that process. But kind of doing the raw heavy to light intensities, these ratios are like kind of way slimmer, kind of much kind of closer to it's something implausible where like if a cell were to have divided, of course it has to renew at least half of its histones, like it can't still have 80% light. So yeah, that's kind of that's a bit of a sanity check, but also we have this kind of method for we're certainly not the ones to kind of develop. People who are doing protein turnover studies in vivo have kind of looked at for a long time.
Hey, Andrew, very beautiful work. I'm just curious. So you listed like one representative protein, HSP9, if I remember right. It's like the
Yep.
degradation contributed a lot to the measurement of abundance. Just wondering, have you I'm just curious, have you summarized like global proteome level, like how how the degree of uh, measurement difference contributed by degradation?
Yeah, certainly. So I didn't kind of get to everything, but yeah, so like these kind of correlations don't kind of provide the full picture. Of course, you kind of still have to look at the magnitude of the trends. And so, of course, there's kind of two ways to answer this question. One is like the kind of magnitude of regulation within a cell type, and like kind of these numbers are going to sum to one. So, within a cell type across kind of the proteome, we see that some cells rely, much more heavily on degradation and like less so on translation and mRNA. At the level of kind of average protein across cells or kind of within a specific tissue, you can kind of look at can look at these different magnitudes. So kind of this gives you a sense, like for this given protein in this given cell type, you'll have, something like 80% degradation. You can kind of look at the average of that across all cells for kind of the relative control. And generally what we see is like, depending on the cell type, close to like somewhere in the order of 20% on average, like control of relative changes by degradation. And, but I don't have kind of the quantitative plots for that. But yeah, certainly a good thing to look at on the relative level as well as absolute.
So we've got one more online question, and then I think we're all ready for a coffee break. So it says, did you measure the percentage of dead cells in your cell suspension? Does it affect your measurements?
Yeah, that's a really good question. We actually had an entire article that arose spinning off that issue. Originally, when we were measuring these cells, we had like a weird subcluster that I was analyzing for months and like thought something interesting was going on. And then later realized that part of these cells from this kind of dissociation were kind of their membranes were permeabilized and they were leaking proteins. And so when we're sorting the cells, we stain them with like Cytox green and kind of do a negative stain for any cells positive with Cytox green to ensure that we have kind of all intact cells. But this is a really important thing to watch out for. And if you're doing a sample prep that doesn't have fluorescent capabilities, we made like a tool to detect dead cells. Essentially, they like leak smaller proteins and cytoplasmic proteins. And you can find that on our kind of SCP Slavov lab and our GitHub.
Thank you, Andrew. So we are ready for a break, but I can't resist saying one thing that relates to a question that Andrew received and that we are going to hear more throughout the conference. That while it has taken a couple of months for Andrew to acquire this data, you're going to hear in the course of this conference several talks that are going to showcase technology that can make it a couple of weeks or even less. So we should all keep in mind that what we are showing here is capabilities that we had a year ago when we started doing that project, and I think for a lot of, and that is true for a lot of the projects across the board. This is a field that developed so fast that in some ways, half of the price we're not yet in the bioRxiv. It's starting to age. It's wonderful that we have conference where the technology forward and is going to speed up progress. So with that, I'm very much looking forward to talking with you and enjoying some coffee and snacks in a short break. And we will return at 11 o'clock for our second morning session.